Hello, everyone, and thank you for joining us today. My name is Geraldine, and I'm going to be your host. We're here to celebrate the release of GATK4, which is a project that has been long in the making. So we're really excited to be here today, streaming live from GATK HQ at the Broad Institute here in Cambridge, Massachusetts. And when I say we're doing it live, I'm not kidding, because the release is actually going out now. Our engineering team had a very exciting morning. I say exciting, putting the final touches to the release itself and to the software. They pushed the big red button sometime around lunchtime, and now the release is going through the pipe, going through the build system. There's a lot of automated testing and so on that gets run. But the thing is, we're expecting the release to, the actual software package to come out any minute now, so it's pretty exciting. As a result, I will be taking over for David Roazan, our lead developer on the GATK4 engine, who's babysitting the release process right now. Anyway, we have a pretty exciting lineup for you today. We are joined by a number of members of the development team of the Broad's Data Sciences platform, and also a really exciting lineup of external guests who have taken a time out to join us today to talk about how we've collaborated and how they have contributed quite a bit, enormously really, I should say, to making GATK4 what it is. So we'll have 4 sessions where we'll talk about different aspects of what's new in GATK4. We'll talk about performance. We'll talk about platforms for running GATK. We'll talk about its expanded scope of analysis. And finally, we'll talk about how we are trying to make the GATK best practices workflows easier for everyone to access and run in the research community. So with that, I'm going to start by talking about the performance improvements in GATK4. Like I said, I'm taking over here for David Roazan, who's the lead developer on the GATK engine, because he's babysitting the release. If we get the news that the release is out at some point, we'll interrupt and have a big cheer. So in the meantime, let's go ahead and talk about the performance improvements that GATK4 brings us. Okay, performance features. Now, the GATK itself has been, for this version, completely re-engineered from scratch to break through some of the bottlenecks that we had with previous versions and to be able to scale up analysis to the levels of scale that we need and performance that we need to tackle the flood of genomic data that's coming our way now. In addition to these improvements to speed and scalability, the engineering team has also put a lot of effort into making the engine more flexible so that it supports more different types of analyses. And some of the GATK developers today will talk about that expanded scope of analysis and what are the new types of analysis that GATK4 supports that we could not do in previous versions. And we'll talk a little bit about the best practices workflows. But for now, let's focus on GATK engine itself and the performance improvements in this new version. And I should say also that and this is a very big deal for us, GATK4 is completely open source under a BSD license. So anybody in the world can use it, take and modify it, and we hope that this will accelerate the pace of research and development in the field. Okay, so let's talk about that new engine. When I say engine, I should explain that the GATK is basically composed of an underlying engine that does all the things like accessing data and so on, and managing a lot of the work that the tools do. And then on top of that, you have individual tools that do specific things. And then you can take these tools and chain them into pipelines. So a lot of the work that has been done to make GATK faster and more scalable has to do with the engine itself, and that's the piece that was entirely rewritten from scratch. That's why it took us a while to get it out to you. I'm not going to go through the laundry list of features, but I'm going to call out a few things that have been especially valuable, and many of these things have been contributed by external developers. including our collaboration with Intel has yielded the Intel Genomics kernel library, which accelerates a lot of processes within GATK. Scalability through the use of Genomics DB, we'll talk a little bit about that. We have Apache Spark support for much more robust parallelism than we had in the past. And we also have now the ability to support cloud features that make it a lot more make it a lot easier to deploy GATK on the cloud. Now, to be clear, you can still use GATK on local machines, but we want to make it easier to use it on the cloud as well. Okay, so to pick just one of these features out of a hat, let's talk about NIO. NIO is a feature that allows GATK4 to talk directly to Google Storage, and it allows us, so in the normal way of running GATK on a cloud platform, or on any platform really, you need to have the file available on a hard drive locally to be able to compute on it. With NIO, we have the ability on the cloud to just, instead of having to localize, which means copy the entire file to the local machine where we want to do the computation, we have the ability to just retrieve a tiny slice, whatever little slice of the file of the data we need to do for any particular operation. This is especially useful, of course, when we're parallelizing execution. And so this is a feature that gives us more speed, also gives us a lot of gain in terms of cost. The pipelines are a lot cheaper now to operate because of this feature. Now, the other big thing that has been improved in GATK4, one other example of a big performance improvement, it has to do with the join calling step in the germline best practices pipeline. This pipeline normally has kind of a bottleneck in the sense that the first part we're doing calling variants with haplotype caller in GVCF mode per sample, and that scales really well. That's pretty linear. But then we have this step of having to consolidate the contents of GVCF files before we can do the joint genotyping, and this is for performance reasons. In GATK, so in this step, in GATK3, with the traditional combined GVCF consolidation, on one subset of sample that we were doing testing on last year, for 3,000 whole genomes, it took us 6 weeks to do the join calling through the traditional workflow. With the GATK4 improvements, you can see that with Genomics DB, which was contributed by Intel, we were able to do the full version 1 of the Nomad dataset, which includes 15,000 whole genomes, which is an enormous amount of data, and we were able to do that in just 2 weeks. So that represents a 5 times increase in scale, and that was processed 3 times as fast. So it's a really big improvement in terms of how this workflow can scale. And right now, we're working on a new call set for the next version of Nomad, which includes 76,000 whole genomes, which would never have been possible with the previous versions of the tool. So that is, oops, sorry about that. So that kind of gives you a taste of the types of improvements we have in GATK4. Now, rather than just talk through myself the technical details, I'd like to invite some of the members of the development team, as well as some of our external guests. And we're going to have a discussion panel where we're going to talk about some of these features and how they're meaningful. So I'd like to invite to the little discussion panel area a couple of guests. I'm going to invite Laura Gauthier, who is a lead developer for GATK. Lead developer for GATK on the germline tools. Paolo Narvaez. Yes, please feel free to clap when the guests come up. All right. So Paolo is a senior director of engineering at Intel's Data Center Solutions Group, and he's responsible for integrated solution development across industry segments, including genomics and imaging. And they'll also be joined by Jean-Philippe Martin, who I'll call JP, JP is with Verily Life Sciences. He's a senior software engineer focused on optimizing genomic workloads on the clouds conveniently and at scale. And we'll also be joined by Marty Lurie from Cloudera, who's a principal systems engineer at Cloudera, who applies Cloudera technologies to solve problems across a wide range of industries. Thank you. And I realize I'm supposed to do this in 2 separate panels, but I'll have you guys sitting for the entire time, if that's all right. Thank you for playing along. All right, so I want to start, Paolo. You've done, you and the Intel team, there's a lot of people behind that, have done a lot to get optimizations into GATK4. And I was wondering, kind of all the really cool optimizations that you guys have put in there, which one is your favorite and why?
Yeah, so I think one of my favorite ones, we learned a lot about how to optimize genomics in general was the work we did with GKL, Genomics Kernel Library. In particular, there was a very interesting kernel called PRHMM, which is used a lot in a lot of different parts of the pipeline. And it was a very fun exercise because we optimize it and optimize it and optimize and re-optimize it many times. As the hardware evolves, we got to re-optimize it for new features that came in the hardware. We then optimize it for FPGA. So we ended up seeing how the same problem can be optimized in different ways for depending on the type of hardware. So and in some cases we saw 100x, 200x increase in performance, and I think it became kind of a textbook example of how you can really parallelize something when you have the right hardware in place.
Thank you. That's fantastic. It's interesting because it seems like just a waterfall, it just keeps coming. It's great job security for all of us, yes. Right? But yeah, it's really great to see all these improvements and the improvements for new hardware as well. I also wanted to touch a little bit on the genomics DB improvements. And Laura, you've been on the receiving end of what comes out of the pipeline. Could you talk a little bit about what that kind of scale, the possibility of that kind of scale affects the scientific results in terms of the analysis?
Right, so at the Broad, we work with data on a variety of scales. We do some small sample patient-specific call sets, and we do some very, very large call sets. So the MacArthur Lab puts together the Genome Aggregation Database, also known as the GNOMAD resource. That's what Geraldine is referring to with those giant numbers on the screen there. And people ask sometimes, well, like, how many samples do you really need? And the goal is to just keep adding more samples because we have some populations that are underrepresented. For example, there aren't a lot of Middle Eastern samples in that cohort right now, and so we can't learn a lot about them or compare patients to the standard allele frequencies for that group. And also, just the more data that we aggregate, the more we learn about the mutability of genes and how important they are in the genome. So it helps further genomic science in general.
So you talk a little bit about this. Now, I like talking about these ridiculously large cohorts because they're just really cool, impressive numbers. But it's true that not everybody drives a monster truck, right? So how does this apply also for more mid-size, like smaller and mid-size cohorts?
So certainly the performance is very comparable for smaller and mid-sized cohorts, but the benefit that you'll get is that you can have a consistent pipeline across all sample sizes, whereas in the old GATK3 best practices, you might be running a hierarchical merge of various groups of GVCFs with combined GVCFs running through it twice, and then once you get to a certain number, you can't do that anymore even. But with Genomics DB, you can use the same workflow for any size cohort, which is a big benefit for consistency.
That's pretty cool. We definitely appreciate, especially from the support side of things, we definitely appreciate when it's easier for people to do things consistently and reproducibly, because it's just fewer failure points. So do you think, and it's either you or Paolo, do you think there are other applications of Genomics DB where we can use this beyond just the joint calling problem?
Yeah, so the original application for Genomics DB was the joint calling problem, right? And so we optimized the data, we created a database and developed it in order to essentially take all the data and process it at once, right? And performed very well and did exactly what it was meant to do. But as we discussed with more applications and more users of it, we realized we could make a general database out of this, and it's not just useful for cases where you need to process data from the beginning to the end, but you can actually use it for random accesses, and it just becomes a general storage for any kind of variant data across multiple samples. So a lot of the effort that is going on right now in development of GenomicsDB is really focusing on the speed up of random access so that you can use it as a general database for any kind of variant storage for any program.
Excellent. So I guess that's, since you're talking about random access, it kind of makes me think about JP's work with NIO and making it easier to just access the parts of data that you need without doing a lot of heavy lifting. JP, you did the work that allows GATK4 to read any piece of data out of Google storage, which makes our pipelines a lot cheaper. I was wondering, like, what motivates you to work on that problem, and how does that fit into the bigger picture for you?
Thank you. Yes, so my motivation is to be part of bringing forward the future, to make it easier to access data. As you explained in the slide and just now, a very nice benefit is that now you can run GATK and access just the part of the data that you need, so this saves you both time and money. Another thing I like is that it's very easy to use. You just, in the command line, you just put a GS to indicate the files on Google storage. There are some other benefits of moving from the old world of running on just one computer and copying the data to this new world of running on the cloud, and you can access the data from the cloud directly. And one of them is the simplicity of this system, and also the interoperability. So because the data is now very large, it makes sense to ship, instead of downloading the data to the researcher's computer, it makes sense to upload the researcher's program to the cloud so it's closer to the data, we can work faster. But with NIO, you don't have to necessarily do this. You can do it the old way or the new way, whichever you prefer. You can start developing on your own desktop computer, and then you can run on the cloud, or you can mix and match software that runs on the cloud and accesses cloud data with some software that runs on your desktop and still also can access cloud data. And the last thing I like about this is it's an open API, and so it's possible for other cloud providers to also do this so that everybody can access data from anywhere. And that's really the vision. And so if you'd like to help with this, my little call to action is, the people who provide data, if you're a researcher, you're sharing data, make sure that your data is available with a kind of traditional file API. Put it in a Google bucket or the equivalent thing for the other cloud providers. And if you're a programmer, if you're writing tools, be it part of GATK or not, consider using NIO or similar technology to allow your users to read the data directly from these cloud storage. Thank you.
Yeah, and since we're doing the call to action, I would say that we're also, as part of making GATK4 open source, is we're hoping that other tool developers will be interested in potentially using the GATK engine itself to take advantage of features like that have already been built in, because it takes a significant amount of engineering effort. It's not trivial, and it's not something that's a comp bio is necessarily equipped to do, but you can absolutely take advantage of what has already been done for the GATK4 engine. And I think that has potential for accelerating the pace at which we can have features like that be usable.
That's true, I agree, this is great. And in fact, both sides of the NIO interface are open source. So the part in GATK, but also the part that's in Google Cloud, the API itself is open source. So if you need to, you can see how it works.
Fantastic. Marty, I would like to ask you, since in your job at Cloudera, you see people from a whole range of industries, right, who are kind of moving to cloud and harnessing the power of all this new tech. Would you say that there's anything special about genomics necessarily as an application that makes it especially suitable, or is it actually typical? And if so, can you predict the kind of gains that people will get by moving to these new paradigms?
Yeah, so firstly, I want to say thanks for, it's great to be here. Tom White, who has done a whole lot of heavy lifting on GATK4, is on sabbatical, so I get to sit in for him. And I hope he's enjoying himself, because he certainly earned it. The parallelism of genomics is very similar to the parallelism that you see when Marks and Spencer does things in retail, Navistar does things in trucking. We actually don't have to leave the medical industry. Cerner is doing some very fascinating things around parallelism to minimize the risk of infection. I also work with a group called DocBox, not Dropbox, but DocBox, and they store every vital sign from every instrument in an ICU for every patient, every second, forever, which is a lot of data. So the benefits that you see, if you think about it, the human genome in 2000 was about $100 million. The human genome last year cost about $1,000. And I believe that last year, 2017, was the first time there was a clinical trial of a vaccine that targeted a specific patient's cancer, custom medicine. So the benefit to researchers is they can get to their data. They can do this on-prem if they happen to have a whole lot of small computers. They can instantiate in the cloud. But there's a greater level of accessibility. And as you've also seen in the earlier slides, tremendous increase in performance.
Fantastic. Well, thank you very much, and please transmit our thanks to Tom White, who has done a fantastic amount of work to help us bring those kinds of improvements to researchers. Now, I'd like to open, if we have time, I'd like to open it to questions from our audience, our online audience only. We're not doing the room audience today. You guys can ask us questions afterwards, but we'd like to give the opportunity to people who are at home and not able to join us to ask questions if there are any.
So we haven't yet had any questions on Facebook.
No questions.
Might have been, maybe people are waiting to ask questions, but I did want to announce that GATK4 is live.
Woohoo! GATK 4.0 is out! Yes! Fantastic. Thank you for that. We've been waiting for that for some amount of years at this point, so that's... Whew. Anyway. Okay. My work is done. Oh, hi, guys. We can handle the rest. Okay. If there are no more questions, then we'll... how are we doing on time? Because this is live, so we're just kind of winging it. Are we doing okay? What I mean is, can I keep this panel talking? Fantastic. We're early on time and everything. So, Paolo, since you were talking about, you told us about GKL, and you told us that you've been doing some testing with the FPGAs. What's the status of that? Can we expect some accelerations for FPGAs?
Yes, indeed. So the philosophy we have for acceleration is we want to. There are multiple types of hardwares, and you can't expect everybody to have exactly the same type of solution, right? So, and at the same time, you don't want to expose all the difficulties, all the complexities of new types of hardwares to the end user. So, what GKL does is provides an abstraction layer in between, where you make a call to Smith-Waterman or to compression or to PRHMM, like we talked about, and it automatically detects what kind of hardware is underneath it, and loads the right type of code, right? So if you have a processor, an older processor with SSC and not AVX, this is pretty ancient at this point, but it will load SSC and not AVX. If you have something that has AVX or AVX2 or AVX3, it will load the right type of code. And if you want to go to the fastest possible acceleration, then you use something like an FPGA, where you're not just using accelerators within the CPU, but using a hardwired accelerator in an FPGA card. And for very parallel kernels like PRHMM, we've seen hundreds of X's improvement in performance. And just as the nature of the algorithm and how well it parallelizes when you have dedicated hardware.
That's fantastic. I heard that Google Cloud is getting some Skylake processors, a new set of Skylake processors. I believe there are some accelerations specifically for Skylake in the GKL.
Correct. I think you guys do have Skylake processors available in your systems, and with Skylake we have a wider vector unit. This is the AVX unit, which goes to 512 bits, which means you basically can do 512 bits of operations at once. So whenever you have the same data doing the same operation again and again, you can really exploit these vector units. And Skylake has twice the width of the previous generation Broadwell. So basically, you can get twice the performance. Same frequency, same number of cores, but you're just doing twice as much. You're packing twice as much data into a single operation.
Great. Well, I certainly look forward to accelerated numbers. There will be some benchmarking efforts. It's a very common question. People ask, like, what are the benchmarks and so on. And the team at Intel has done quite a lot of benchmarking effort, and we'll have some of our own, so that we can kind of give people an expectation of, depending on what their hardware is, what kind of performance they can expect from the new versions. All right. If there's anything you guys want to bring up, or we can discuss, but otherwise I'll thank this panel for a very informative discussion. Thank you very much for joining us. It was really a pleasure to have you with us. And we will move on to the next session, which is going to talk about platforms for running GATK. So first, I want a round of applause for our guests, please. Thank you very much. Thank you. You can tell I'm kind of new at this. I'm probably not going to be hosting the Golden Globes anytime soon, or the Academy Awards. I watched a few videos of Neil Patrick Harris doing some amazing opening numbers. It was inspiring. I don't think I'm quite ready for the show of singing and dancing, but what I can do is queue up the next moderator, Danielle Ciofani. Thank you. Who is going to lead this session talking about platforms for running GATK. And with that, Danielle, I will cede the floor.
Thank you. Hi, everyone. I'm Danielle Ciofani. I lead data strategy and alliances here at the Broad. And I'm here to talk to you alongside our technology partners about how we're making it as straightforward as possible to run GATK on any infrastructure. So we'll talk a lot about how we're optimizing for on-prem and cloud computing, but I want to call out first and foremost that an individual user can run GATK on their laptop today using local hardware. If you want to string together pipelines or workflows at scale, we've developed our workflow description language WDL, as well as our execution engine Cromwell to build pipelines. Cromwell, of course, will execute on an on-premise compute cluster or against the cloud. Regarding on-prem, I just want to take a moment to acknowledge the hard work from our Intel collaborators. We've worked closely with them over the years, optimizing every part of the pipeline holistically, and you've heard a little bit about that already today. But as part of their commitment to ease of use, Intel's built this rather turnkey solution that incorporates the entire pipeline execution toolkit, including but not limited to GATK, WDL, and Cromwell. And they've organized it in a stack, if you will. They call it Big Stack, also known as the Intel Select Solutions for Genomics Analysis. And it's really meant as a one-stop shop for on-prem infrastructure. In terms of how we at Broad have organized our production pipelines, we have sort of a hybrid setup where part of our system is on-premise and part of our system is also on the cloud. Data comes off the sequencer and is routed to a file system on-prem, which is subsequently routing data to the cloud. Our production pipelines leverage WDL and Cromwell talks to the Genomics Pipeline API from Google Cloud, which spins up a compute engine cluster based on the instructions of the workflow. The ultimate output of the pipeline is written to a Google storage bucket, which ends up going to our customers. I want to talk for a moment just about the partnership that we've had with Google Cloud over the years. We've worked really closely to effectively co-develop the earliest generations of the Genomics Pipeline API. And over the course of the past few years, we've really scaled up our ops pretty tremendously. To date, I think we've processed 5 petabytes of data, and in the past year amounted to over 100 million hours of core hours on Intel processors on Google Cloud. So it's a lot of data, and it's working really well. And most recently, we've been optimizing for cost. So since the start, we've effectively reduced the cost of our production pipelines by about 95%, and you'll hear more about that publicly in the next coming months. So I want to talk one moment also about Cromwell and our commitments to interoperability. A couple of things that we'll note, and our technology partners are here to talk to you more about, but Cromwell will soon support interactions with not just Google Cloud, but other clouds. And also, as part of our commitment to GA4GH interoperability, we will support not only WDL, but common workflow language. So that's our production setup. What I want to usher in is actually a bigger conversation around just GATK4's flexibility overall. We run on Google Cloud. We acknowledge that not everyone will, and that's why we've invited a number of cloud vendors here to talk about how they are respectively optimizing for GATK4, as well as potentially adding a Cromwell backend. So without further ado, I'm going to introduce Hershan Lin from Alibaba Cloud. He's in bed right now because he's based in China. So let's give him a warm welcome regardless. He'll listen to this over the phone. All right, let's see. Nope. I'm going to ask for tech support from Geraldine to make sure I can play this.
It's what I do.
Yep.
I think you're supposed to.
Just click. Okay.
Hi, my name is Hershan Lin. I'm the product lead of the Genomics Computing Solutions from Alibaba Cloud. It is my great pleasure to join the community to witness the launch of GATK4. During the past few months, we have been working closely with Bro's development team on getting Cromwell and the GATK run on Alibaba Cloud. I'm going to give a brief introduction about the work we have been doing and the experience of running GATK on Ali Cloud. So here's the overarching architecture. The GATK pipelines are written in WDL, which are interpreted and executed through the Cromwell workflow engine. Cromwell has a nice modular design to facilitate adding a new executing environment. What we need to do is to implement a new backend for executing commands and a file system for storing the meta and application data. We implemented the backend by leveraging the batch compute service on Alibaba Cloud. Batch compute is a distributed computing service that allows users to easily execute a large-scale batch processing tasks. Users can deploy applications with Docker image and invoke a command with a simple API call. For data storage, we implemented a file system on top of Ali Cloud's Object Storage Service, or OSS. OSS is a secure, scalable storage service perfectly for storing genomics data. Putting things together, when the Cromwell engine processing the GATK pipeline decides to run a command, it submits a job to the batch compute through the backend. Batch compute will then automatically spin up a virtual machine, fetch the necessary data from OSS, process it, and then write the results back to OSS. One challenge of running bioinformatics tools on the cloud is that these tools are typically written to access normal file system, while object storage has a very different interface. One way to resolve this is to copy the necessary data to the local file system of the virtual machine, but this will take more disk space and incur extra overhead. To address this issue, we leverage a special feature of batch compute, which can directly mount OSS objects to the local file system of the virtual machine. By doing so, bioinformatics tools can access data on OSS through the file system interface without paying the cost of a copy. In addition, when accessing an object through the mount interface, the batch compute service will automatically prefetch data to improve the reader performance. Also, when multiple nodes within a cluster are accessing the same file, these nodes will form a cache ring. Consecutive reads of the same data will be served from the cache without going back to the OSS. This caching feature can greatly accelerate the distribution of shared data like reference genomes. We heavily use the mount feature through the Cromwell implementation. We also provide some runtime attributes specific to Ali Cloud's platform. We support the spot instances, which allows users to name the price of the resources they would like to use. We also allow users to use the reserved cluster in batch compute, which will save machine startup time when running a large number of small jobs. It also makes the debugging much easier. Users can use the mount feature to access the object storage, as we mentioned in the previous slide. Okay, so how do we run a GATK pipeline on the Alibaba Cloud? In order to make it simple, we offer a pre-configured environment. We deploy the Docker image and the common data such as reference genome and indexes in the public OSS buckets. We add a Cromwell app in the batch compute, which can interpret the Widow scripts and launch jobs accordingly. It takes the three simple steps to run a GATK pipeline with the batch compute CLI. First, you can use the CLI to generate a pre-configured GATK pipeline template, which is defined in Widow as distributed by Broad, but with runtime attributes tailored for the Ali Cloud environment. Then you can specify the input data by modifying the input section of the Widow. And finally, you can use a simple command from the CLI to run the script and submit the jobs to the batch compute. The Cromwell and the GATK support is available only in China regions for now. For more information, please go to the product page of Batch Compute. I would like to mention that as a founding member, Ali Cloud has been working with Intel, BGI, and Broad to establish a Chinese GATK community. We look forward to providing GATK4 to all the users in the Chinese community very soon. That's all I wanted to say. Congratulations on the launch of the GATK4, and I would like to thank Broad's GATK team for all the help along. Thank you for listening.
All right. Next, I'd like to welcome Angel Pizarro from Amazon Web Services Research and Technical Computing. Angel leads the global genomics, life sciences, and precision medicine initiatives within the Amazon Web Services Research and Technical Computing team. Thanks.
Thank you.
All right. Thanks everybody for inviting me. Thanks, Danielle. It's probably a fair statement to say that outside of the Broad, the majority of GATK workloads actually happen on AWS through no reason of us, right? It's all you guys that made it happen. So what my talk today is, it's short 5 minutes, but it's about GATK on AWS. How would you actually take advantage of it? As a new user, as a current user, you might have some questions, especially with some of the changes that's happened underneath the hood. So I titled this Some Assembly Required. Because if nothing else, there's a lot of options in AWS for running GATK from single node, where you can create your bash scripts, SnakeMake, or use other workflow systems like SnakeMake or Nextflow along with the CLI tools. I'll cover Cromwell in a second. Just, it's not put on here for a particular reason. You can also create, high performance clusters, what you think of as Beowulf clusters today. Predominantly, this is actually how most people start out. Even through moderately sized 70,000 whole genomes that they analyzed, just using traditional schedulers like Slurm or HD Condor with file systems like Lustre, Intel Lustre, or Weka.io, or others. There's also other clustering solutions on AWS, and I'll cover those in a little bit. And then, honestly, let somebody else do the work for you is really how we think is if your goal is to do science and analyze samples, having somebody else like either the Fire Cloud on our good partners or other partner platforms that are on top of AWS, have them run the workflow for you, that's what you should do, because your time is better spent squishing cells. Right, so genomics platform, Seven Bridges did put out this nice blog post on an early beta, I believe it was beta 5 of GATK4, where they took a look at the run times, and this was, at that time, a 30% improvement over GATK 3.6 on their platform, which is quite good considering you guys have done a lot of optimization since then, so I'm actually excited to see an update to that blog post by them and other folks. DNA Nexus themselves have written a tool that imports WDL workflows into their own pipeline, and they can work and do GATK4 today, which is pretty exciting, right? So again, and same for Illumina and other sorts of folks who have done an implementation or a translation of a best practice pipeline and implementation on their platform. Again, let other folks do the work for you. But if you're hard-nosed or if you really want to try it out yourself, running on a single node on AWS is as simple as a bash script. This is a snake-make script here, but bash is fairly equivalent. Why this works, and particular to AWS, why this would work even on a single node, is that there are some really large amounts of compute available to you, even within a single instance, from 4 terabyte RAM systems, which are quad socket CPU servers, to 8 Xilinx FPGAs, really large compute, and Skylake X with what Paolo was mentioning earlier with the 5X, with the 512 AVX registers, and including our new instances have and this is neat from a technology perspective, but might be way too much information for this audience, but we've developed our own hypervisor that's a hardware accelerated hypervisor that allows you to do some really neat things with containers that we've started to release in the coming years. So that's going to be pretty exciting how people leverage this new hypervisor. But to get down to the specifics, how do people actually do it? Basically they use object storage, and this is what happens underneath the hood with Cromwell as well and other systems, is that there's a data staging to some sort of compute, and then you get like Rihanna and start work, and then you go back and upload your results either halfway through it or as it goes through. You can do that on a single instance. You could do that on clusters with parallel NFS, again, staging from Amazon buckets. You can create some orchestration services with a bunch of other things that we have tooling, but honestly, I'd rely on workflow systems like Nextflow, like Cromwell, like other things that are coming to really implement this for you. This is more if you're really interested in digging down very deep into our infrastructure stack. But there is we did do a workshop on this. And finally, for Spark, when you're doing joint variant calling across those tens of thousands of genomes, for the time being, and again, I'll talk a little bit about this, but HDFS is your friend. So you can import data at scale extremely fast through a managed Hadoop cluster and then do all your work on HDFS and checkpoint back as necessary. So what about Cromwell? If you were at ASHD, I might have sent this around to a private group Slack channel. This is a nice little lazy river that was at the hotel that we quite enjoyed. And at that point, we were here where we really came to agreement to go forward with the Broad to create a Cromwell backend that's native to AWS. And then all of this over here was some contracting. And we're about right here right now. So stay tuned. I think in a few months, we'll have some pretty decent results to show you because we just, we've gotten around the bend of the forest. So I think we're pretty good now. Thank you. I don't think we have any time for questions. Is that correct? Okay, so maybe at the end or something. Thank you, though.
Okay, next up is a recording. Again, this time I think I'll be able to play it myself. This is Ravi Pandya from Microsoft Genomics. He's a seasoned architect of genomic sciences and works in the Microsoft Genomics team on high-performance algorithms and AI for genomic data.
Hi, this is Ravi Pandya with Microsoft Genomics. We've been working for the last year and a half with the Broad Institute to optimize the GATK for the Microsoft Azure cloud. Now, obviously you can just spin up a virtual machine, install GATK and whatever other scripts you want on it, and that will clearly work just fine. But given the importance of GATK as the de facto standard for genomics around the world, we wanted to do more than that. We really wanted to make it easier, faster, and more scalable for people to run GATK in our cloud. So we built the Microsoft Genomics Service, which is a service on Microsoft Azure that you can sign up for like any other. You go to the portal, you search for genomics, and you create a genomics account. Once you've done that and you want to run a workflow, you can basically upload your info files, FASTQ or BAM files, to Azure Storage, and then you install a Python client using pip on Windows or Linux. Then you need to set up a configuration file for each run that describes the inputs, outputs, reference, and any other parameters you might want to give. And then you simply submit that, and this will take care of running it, and we'll take care of everything from then on. We will spin up the appropriate machines, take care of the compute and data movement that's required, and then when it's all done, you just need to go and download your aligned BAM file and your VCF from Azure Storage. You can run this either through whatever scripts that you're currently used to, or you can also work with one of our solution partners like DNA Nexus or BC Platforms. Now, behind the scenes, there are a number of different optimizations involved in making this run. At the very top level, we manage a cluster of machines using Azure Batch and dispatch each workflow to an appropriately scaled machine with the right amount of RAM, SSD, and cores in order to run the analysis from end to end on a single machine as efficiently as possible. Within that machine, we built a customized engine to handle I/O and concurrency as efficiently as possible, and we've taken various pieces of the GATK pipeline, such as BQSR duplicate marking, and rewritten those from Java into C++ for efficiency. And then at the very bottom level, we've taken some core algorithms, such as Smith-Waterman and PairHMN, and optimized those using the Intel vector instructions for maximum performance. So the end result is that you can run an analysis that might have taken over a day in a matter of hours, and you can do this very easily, and it scales up automatically and is available worldwide. We have been really enjoying this opportunity to work closely with the Broad in order to support scientists and clinicians who are using genomics to advance human health, and we're really looking forward to continuing to do so in the future with the launch of GATK4.
Okay, and last but not least, here for the platform section of our talk, we have Dr. Frank Liu from IBM, who has been leading the current work between GATK and IBM since the collaboration started in 2014. Give him a hand.
Thanks very much, and again, congratulations to the official launch of GATK4. As Daniel mentioned, IBM has been an active partner of GATK4 development since 2015, probably 2014, end of 2014, and among the key people, I mean, besides myself, Carlos also sits in the audience, and also have Ying He, who cannot come over here because of family reasons. I mean, participating in the weekly meetings. Thanks very much for the David and Luis for the tremendous effort to not only to get code in place, also to coordinate efforts of such a diverse team. So inside IBM, we have a lot of, we feel very really pleasure to participate in such a big project, given the importance of GATK, the GATK toolbox. And so right now, GATK4 basically runs out of box on all the Power platforms, open Power platforms, including Power 8 and Power 9. We support both worker mode, the container, as well as the Spark, Apache Spark mode. Right, usually what we recommend use is Ubuntu or Red Hat Linux and Little Endian only. IBM is ongoing transition from Big Endian to Little Endian. We support both OpenJDK standard Java platform as well as IBM's home Java JNI, which was open sourced late last year. And also all the accelerated native libraries such as PyHMM are also available on open power platforms as well. And we see very good performance gain compared to native Java code. For the storage side, we support both IBM's own Spectral platform, Spectral Scale, which is formerly known as GPFS. If we run the Spark mode, then we basically support both GPFS as well as standard HDFS storage. For the recommended system we use, we have a few lines of hardware and the Power 8. We usually call it 822LC, usually called Firestone. Also have a more advanced version called the Minskys. And IBM Power 9 just released late last year, and more models will be available later this year. And IBM Power designed this heavy core system, and they have a very big memory bandwidth and large number of cores and a large number of threads, which is very suitable for this kind of big data applications. Usually we recommend people to use IBM Java, which is also open source component. We use NVMe as a scratch storage or use SSD if you prefer. For the worker mode, recommendations use GPFS, and personally I use HDFS, and the Carls use GPFS for Spark workloads. So we put a lot of effort into supporting, investigating the GATK4 on Spark. A lot of effort in the debugging and tuning of Spark configurations to make sure that all the nodes have a properly tuned to have proper performance. And so this is just a nice chart showing the GPU utilization when we're running on the, in this particular case, I believe is 4 nodes and the GPU, very nice GPU utilization, and have to thank the broad team to get all the work done. And also, but also look at a few things that about Spark target JVM level configuration enhancements, for example, garbage collection strategies, optimize just-in-time compilation, and JVM log contention scheme, etc. All these effort, most of them can be released or has been released into open source projects. So I'm hoping that the Bro team can also pick up these and for the future performance enhancement of GATK. IBM also has to work very closely with IBM's clients in terms of using GATK4 to build clinical pipelines for real clinical applications. This is one example we use is workflow description language, and we have worked with partners like MD Anderson and Cloudera to find a way to have automatically generated this workflow description for GATK4 workloads, and also have the functionalities to automatically collect the metadata to build the correct provenance of the data flow. This is very important for the IBM's customers because unlike the scientific research, for the real clinical trials, we really need to have to keep track of all the provenance. I think I'm running a little bit short on time, so just as acknowledgement for the GATK4 team I mentioned before, and also after you can see that we have quite a few people working behind the scenes and collaborating with MD Anderson, Cloudera, and also Washington University. And thanks very much. Any questions?
All right, thank you very much. That was a really important session for us in terms of we want to show that there's there's a lot of options for running GATK. We don't want anybody to feel locked into a particular solution, and that's why we put a lot of effort into working with these collaborations, with these industry partners who are helping us make the range of options bigger. So I want to really thank our partners who have made this possible from all these different vendors. All right, now we're going to move on to talk about the expanded scope of analysis in GATK4. I'm going to invite Lee Lichtenstein, who directs the somatic method development efforts in the GATK team, and is going to tell us what's new, what are the new types of analyses that you can do with GATK4. Thank you.
Hello, everyone. For those of you who don't know me, my name is Lee Lichtenstein. I'm an associate director in the GATK of the GATK, and I'm also one of the associate directors in the DSP methods group. So today, I'm excited to tell you about the expanded scope of analyses. In GATK4, we really tried to increase the breadth of what you could do with the toolkit. So first of all, we started treating the somatic pipeline as a first-class citizen in the GATK4. Here you can see very happy and dapper David Benjamin. Why is he excited? Because MuTech 2 has basically been released live, including a whittle, for production-level consumption. And he's also excited because up there you can see, due to the local realignment, he was able to pick up a 150 base pair indel that he would have missed with a pilot-based approach. Additionally, we've also added somatic copy number. Here you see a very fancy Sam Lee. Here, as you can see, we start with raw copy ratio data that we get from coverage. We can then normalize it several ways, make calls on it, segment it, get our segmented calls, and essentially we can start calling areas of the region as either amplified, deleted, or copy neutral based on coverage. And we also use the minor allelic fraction now to further help with the segmentation. Additionally, the previous version had a bit of an issue that many researchers found when they were trying to run WGS, where we had this hypersegmentation problem. And as you can see here, we would get things like 10,000 segments for a genome, but clearly some of these are the same segment just broken into hundreds of pieces. And it took a very long time to generate this kind of data, and it wasn't very good. So we spent a lot of time in the newest version of the somatic CNV pipeline to address that. And now we can generate something a lot more reasonable in a substantially less time. And here we show we're doing this with 8.6 million targets. So we've broken up the genome into 8.6 million bins, and we're still running it in 11 minutes. GATK4 also has a germline CNV caller, thanks to Murtash, pictured below wearing his best, where we can also take raw coverage, much like we do for the somatic CNV calling, run it through a fairly sophisticated machine learning model, and get integer copy number calls for the germline. And we compared this against some other tools, and for rare germline copy number events, using whole genome as a kind of whole genome calls as a ground truth, calling on exome data, we could show that GCNV basically outperformed other common tools that have been in this space. Additionally, we are also working on a model for calling common events, and the performance is down below as well. And these are constantly being improved, as you can see here, just going from 4.beta to GATK4, the release version. There's already a performance improvement. Sam here, Sam Friedman, here pictured with his work clothes, apparently, has been working on improving our germline filtering of short mutations, SNPs and indels. And here we compare it against some other tools, including what we used to run, which is VQSR, the kind of current state of the art in our pipeline. He compared it against another germline calling tool. And here are results with our initial our initial preliminary test data. And as you can see, we're performing quite comparably to the published results from the other tools. So from there, I think we will start the panel.
Fantastic. Yes, Lee, thanks for giving us this overview. Thank you. Lee's going to join me here for a panel discussion, and we're also going to be joined by two other developers of the GATK somatic methods. David Benjamin, who leads the efforts on somatic short variant calling, MUTEC2, and Sam Lee. We have two Sams. We have too many redundant copies of names in the development group. Too many Davids, too many Sams. No offense, guys. Anyway, Sam, welcome. He leads the somatic CNV developments. So I want to start with you, David. Before you came along, MUTEC2 was kind of languishing without getting any attention in GATK3, and you kind of got the task of dragging it, kicking and screaming into GATK4. I guess I was kind of wondering if it was just kind of you drew the short straw on that one, or was it actually a problem that you found interesting and interesting things to solve there?
MUTEC is an incredibly interesting problem because of the just enormous variety you have. I mean, there's the part of semantic variant calling where you get to assume that all your reads are independent and you believe your sequencer, and then you can build this amazing castle in the clouds Bayesian model. And then there's the part where, well, that's not true at all, and you can't believe it, and then you have to do more open-ended black box machine learning. Then, thanks to the assembly that Christian Sobolskis, the original developer, put in MUTEC, you have to know a thing or two about good old-fashioned algorithms of computational biology. And then you just have to make the thing fast, so there's a big element of software engineering, too. So whenever you want to procrastinate on one thing, you have other work to do.
That sounds like a reasonable approach. Very productive. Well, thank you for getting MUTEC2 into shape in GATK4, because it's something that people have been asking us for a long time. So we're very happy to have it in a ready for prime time version. I kind of want to ask Sam, it's a similar question. You kind of did the same thing with the CNV tools in the sense that there was an existing tool called RecapSeg, and then you kind of took that and ported that into GATK4, then gradually made it better, and now it's a completely different thing. And can you talk a little bit about that process and what you found interesting?
Right, yeah. So, I mean, like doing science, writing software is an iterative process. You start off with the germ of an idea or a prototype of an algorithm, and you write a first paper or implement the first prototype, and you say, well, this isn't working like I expected, or there's more to the story, and you just continually make it better, right? And so even before RecapSeg, which is sort of a predecessor of the CNV tools, there was CapSeg, right? And this was an R script that was developed by some people in CGA, evolved into RecapSeg, which Lee had a big part in developing. We brought that into the GATK with the CNV and ACNV pipelines, and the versions that we're releasing, I think we're calling the model segments pipeline, that's even better. Right? And on the same thing, on the germline side, we started off with Murtash's germline CNV caller. He rolled out this really nice spark implementation. Like, we can do better. We can use advanced machine learning packages like PyMC3 and Theano, and I think that's what's going on in the release. So this process of continual improvement, continual relentless improvement, that's a hallmark of the CNV development team.
Excellent. So what's next in terms of improvements on the somatic sides? David, do you have things on MUTEC2 that you're still tweaking?
Oh, I mean, yeah, MUTEC has not come close to reaching its potential, actually. One thing that people always want in cancer is to solve the needle in the haystack problem of a small signal and an enormous noise. But the demands are actually getting greater. So people are starting to do something called a blood biopsy, where instead of sequencing a solid tumor, you sequence blood, and you get the tumor signal from the tiny amounts of dead cancer DNA that's spewed out into the bloodstream. So your signal has now become smaller, and we weren't exactly done solving the original problem. So small signals are another problem. It doesn't just enable blood biopsy. One thing it'll enable is really getting a refined estimate of the subclonality of a tumor, because if you have this tiny little subclone that's branched off from another one, it's going to have a tiny signal and tiny cell fractions. And it would be extremely great for the cancer community to have a tool that gave you the data to infer that.
All right. So does that also help us do tumor-only calling? Or is that a separate problem? Like it's something that people also ask us a lot. What if I only have a tumor sample? I don't have a matched normal. Can the new tools do that, basically?
So MUTEC can do tumor only. I'd say it does it a little bit better than it has in the past because we've have better germline resources. We have Nomad instead of dbSNP. So dbSNP just says someone at some point thought maybe this was a variant. Nomad says a bunch of very intelligent, rigorous people with a billion samples or whatever think this is a variant. And they know how many humans it occurs in. So you have much more ability to say this is probably a germline variant, even if you only have the tumor.
Yeah, that makes a lot of sense. Thanks. Do we have any questions from the audience?
We do, in fact, have many questions. First of which comes from Gabe on Facebook. Are there best practice guidelines updated for GATK4 for common workflows, individual germline, trio germline, joint calling, etc.? And how different are the command line parameters?
I might take the first part of the question, which is that we have updated all the best practices that are on the website as of last night to reflect the new GATK4 paradigms. Some of the very, very new improvements were still coming in. These guys were still merging code over the weekend, pretty much. So there's still a few changes that aren't reflected in the documentation. So that's something that over the next week or so, we're still going to be buffing up that. But we're already pointing to reference implementations. So while we're still stringing all the words together, you can go and look at the actual script that does the work. And you can see exactly how we run those best practices. And we'll talk a little bit more about the best practices pipelines in the last session today. But in terms of arguments, yeah, there's a fair amount that has changed, right? Or not.
I mean, up to a degree, yeah. We have tried to maintain consistency in the somatic pipelines with the germline pipelines. And in terms of best practices in Whittle, we do publish the entire workflow. Therefore, in theory, you can go to the repo and get a Whittle that should work out of the box and not have to worry too much about how you would specify those command line parameters at all, even if we're changing them out from under you, as long as you have the Whittle and the jar correctly in sync. Yeah.
All right. Great. And along those same lines, John from Facebook asks, what can a typical user expect when migrating their workflow from GATK3 to GATK4?
What can you expect? A lot more speed, more things, more accuracy. I would say that there's one big thing that we've tried to do is make it easier to not have a lot of guesswork to do when implementing the analysis, implementing the scripts. There's definitely some things that have changed in terms of the syntax, the specific argument names and things like that. And we'll have a migration guide posted in the near future to help with that.
Great. Andrew from Facebook asks, do the performance enhancements of GATK4 require Spark? Is there still support for non-Spark parallelism in GATK4?
So I want to differentiate between having a Spark cluster versus just calling the Spark library. One of the nice things about Spark is that you can run the Spark library without a Spark cluster. And what GATK4 does is to get its parallelism, it just calls into Spark. Spark will allow you to get local parallelism if you have a multi-core machine. And for any tool that has a Spark version, this is pretty trivially specified with the Spark master parameter in the GATK script.
And are the tools, like which of the tools in the somatic pipelines use Spark currently?
Currently, the somatic creation of a PON, because that is actually grabbing a lot of data that is using Spark. Our case sample workflows is actually fairly fast, so we actually have not done Spark integration there yet.
All right, excellent. Oh.
That's all we have for this panel.
All right, excellent. Well, thank you very much, Sam and David. Lee, you can stick around. Let's have a round of applause for Sam and David and their contribution. And I'd like to invite other Sam Friedman, to join us, as well as Murtash. So Sam Friedman has been working on using neural networks to improve the germline variant filtering in GATK4. And Murtash has been developing the germline CNV pipeline. Thank you, guys. Oh, yes, and forgetting technically my boss. Eric, why don't you join us on the panel? Eric Banks, everyone. Eric, yeah. Oh, yeah. Yeah, you can go. You're fine. Thanks. Thanks for joining us. Sorry about that.
I forgive you.
Thank you. The next review cycle is only in June, so I should be fine. So, Murtash, we're kind of getting into the very cutting edge stuff. There's a lot in development in GATK all the time, which makes the job really challenging, but also interesting. But you guys are working on very new developments using some pretty new technology. So, the germline CNV, it's pretty close to being released, right? It's not quite ready in 4.0, but when can we expect to be able to play with your tools?
Yeah, so, well, at the moment, the germline CNV calling tool is released as a beta feature in GATK4, which means that people can already start using it. But what I'd like to say is that this tool has been undergoing significant development in the past 18 months, and we have changed so many things about it, about the internal model, about our approach to inference of these models, the methodology, all the infrastructure. They have changed so many times. We have continuously improved it, as I hope the plot that Lee showed you, it shows that. But at the moment, it's the core computational part of it is pretty stable, and you can take it for a spin and just try it out and give us feedback. So what I'd like to say about it is that I'd like to say that the germline CNV calling looks like a Tesla Model X. Okay, so it has a pretty fancy solid engine, but maybe the LCD is a little bit broken, and the power steering maybe doesn't work. So we're getting there. But you should definitely take it out for a ride, and then just give a feedback, see how it works. There are a number of issues that we're working on. We would like to improve the output format, for interoperability with downstream analysis tools, and best practices guidelines. Better default values for different data modalities, for whole exome data, for whole genome data, gene panels, and these are all set to be done this quarter, and we will basically just give it to the users in a maintenance release. So just, I guess we'll keep the users posted in GATK forum.
Cool, excellent, looking forward to it. So the tools use machine learning to do their magic. It's a little bit magical to me, but I wanted to know, what prompted you to move towards machine learning rather than more traditional approaches?
Well, I mean, in a sense, all the tools in GATK use machine learning. It's just that for some reasons, we needed to use some of the more fancier machine learning developments. So in a sense, both the somatic and the germline CNV calling tools in GATK, they were born out of our frustration with the level of the tools that existed for read depth based CNV calling. I remember actually one day the frustration in David's eyes is, none of these tools have a solid model that is inspired by the intricacies of library preparation and sequencing technologies. So we sat together, we put together a model. The model got fancier and fancier and more complex. And at some point, it got so complex that we just couldn't put together a solid mathematical inference algorithm on a few quiet afternoons of just scratchbooking and just long work. So we decided that, okay, we're going to embrace the same division of labor that exists in the industry today, that as method developers, it is in our best interest to basically use some of the more sophisticated infrastructures that has been developed for deep learning applications, stuff such as computational graphs and black box inference engines. So it was a technical tour de force, but I'm glad to say that I think at the moment the GTK CNV pipeline and its results are probably best in class.
Nice, very nice. Well, congratulations on that. I want to see some benchmarks that we can share with our users, right?
Absolutely.
They want to be convinced. Sam, we're kind of talking about pushing the envelope here, and you're definitely pushing it with the neural networks. Speaking of things that going to cause us headaches on the support team to try to understand what the hell is going on in there, so that we can even explain to people. I guess my question on that front, though, is more like, what was the driving motivation for tackling germline filtering with these tools? Like, isn't germline short variant filtering kind of germline short variant discovery is kind of a solved problem?
Yeah, I think when it comes to SNPs, we're doing a very good job, and pretty much all the callers out there are doing a very good job. When it comes to indels, there's still definitely some work to do, and it really depends where you're looking in the genome. So as we sort of expand our knowledge and our ability to sequence, we're finding areas that are harder and harder, and indels that are harder and harder to call. Why we picked germline for using deep learning is because we had lots of validated samples with VCFs that we trusted that we could train from. So the big challenge is to find a lot of data that we can use to train our models. And for germline, there were these good validated VCFs that served as training data, where it's a little bit harder to find training data for the somatic samples. So I think it was a confluence of there was still improvement to be gained, and we had the data to train with.
That makes a lot of sense. Do you think we'll be able at some point to make the jump to for example, organisms, non-human organisms that don't have that kind of training data? Like, what can we do to tackle those use cases?
Yeah, we've already seen that our models generalize to other mammalian organisms to some degree. I mean, there's definitely less generalizability across species and across different platforms of sequencing technology, but there's something we're learning in these models that does generalize. So we can start focusing in on that if it becomes a big concern to expand to mice and.
Well, it's definitely a big thing, and it's not just mice. And there's other like mammalian species, but there's also agricultural, like crop research, where you have exotic ploides and things like that. So we're really looking forward to seeing how those improvements translate into those typically very less kind of underserved areas in genomics. Eric, you were one of the original members of the band, right, of the GATK development team. I'm not going to say.
I think you're calling me old, right?
Yeah, well, you've been around forever. And I think you and David Roazan as well, right? Maybe the last original members still in the GATK development team. So how do you feel about how the team and the toolkit kind of have evolved over the years? You're less involved in development itself now, but how do you see the young ones? Huh?
Right. I'm a bureaucrat.
Yes, yes.
No, it's a great question. Actually, with the hoopla around this release, I've been thinking about the early days of the GATK. We started developing it in 2009, quite a while ago, for the 1,000 Genomes project. And I remember, when we first realized you needed to do post-filtering of call sets, we had some hard filtering. There's a tool called variant filtration that I think still exists, where you have hard cutoffs. And I remember David Altschuler, who was a visionary in genomics and one of the leaders of the 1,000 Genome Project, sitting down with us and saying, "This is not right. You'll have a SNP that in 9 out of 10 dimensions looks perfect, and in 1 dimension just barely fails, fails your filter, and you're filtering it out. And you'll have a SNP that looks pretty cruddy for most dimensions, but just barely passes the filters, and that's in there. This isn't right. You guys are data scientists. You should be smarter than this. You should do something better." And we did something better. It was the first machine learning tool in the GATK. At the time, it was VQSR, Variant Quality Score Recalibration. So I know Murtash mentioned that all the tools in the GATK use machine learning. It wasn't the case then. It was just the first shot, and we were very proud of ourselves, and it was great then. But over the years, I've just seen the geniuses here and other panels and the rest of the team just approach it with rigor and an impressive amount of math and hardcore computer science and probability and statistics, and just made the tools so much more impressive. And I'm excited to say, this is a brave new world. I cannot wait to see what they're doing next.
Me too, me too. Thank you. Do we have any questions from the audience, from our live Facebook audience?
We do. Rayoni of Facebook asks, how is the implementation of deep learning algorithms in GATK4 going?
Going well. So there's a bit of a language barrier in that a lot of the great new machine learning libraries have been written in Python or C++, and the GATK is a Java tool. So we've done a lot of engineering work to allow us to sort of seamlessly deliver the same nice Java interface to our users, but secretly in the background using some Python to take advantage of the latest abilities of auto-differentiation and neural net building that's available there. So it's been a big challenge, but we're making great progress.
Great. Question from Wim on Facebook asks, will there be germline structural variant calling tools in GATK4 for discovering/genotyping large indels, inversions, and translocations?
Yes, it's actually, I'm not developing it. I'm, as I say, just a bureaucrat. But there's a team working on that. You know, none of them are on this panel right now, but they're developing it, and they're making some impressive progress in that.
Do we have an ETA or just general roadmap timeline?
I mean, structural variation detection is a very hard problem. All these problems are hard, but this is very hard. We're hoping to get something out early this year for the users as a release.
Cool, thank you.
Perhaps time for one short one. Does GATK4 include indel realignment module? It was not in GATK4 beta.
No, it's gone. It's gone. It has been swallowed in the dust of history. Yeah, I mean, if people want to resurrect it in their own fork or something like that, or even contribute it back, we can consider it. But we don't. There's a pull request right now. Oh my gosh. Well, we'll talk about it. Maybe there'll be like a shadow distribution that has all the things that we don't want to have to support, but that people want anyway. We're looking forward to the future and the new tools that you guys are building. That's pretty awesome. Thank you so much for taking the time. All right, and now we're going to move to the last session in today's event. And Ruchi Munchi, who is a software engineer in the data sciences platform, which is the wider group that the GATK development team is a part of, is going to tell us about our efforts to make the best practices easier to access and deploy for the user community.
So I'm here today just to talk about the GATK best practices, in case you haven't heard of them yet, and also where to find them, the most important question. So in general, the GATK best practices are step-by-step recommendations on how to perform variant discovery on high-throughput sequencing data. So there's a few different flavors of best practices pipelines available as of now, based on the kind of data you're using and the kind of variant discovery you're interested in. One nice thing about the best practices that the GATK provides is that they're filled with documentation that describe the key principles that are involved in the processing and analysis of the data as it's being transformed from raw reads coming off a sequencer all the way to a filtered variant call set. And another plus of the best practices is that they're validated and tested against real data. So they're pretty good to go out of the box. So one place you can find them as of today is a GitHub repo, GATK workflows, and you can see all the different flavors of pipelines available here. And they include some inputs JSONs you can use right off the bat that contains sample data, as well as the actual workflows. So a little bit about those workflows. So GATK has taken a reference implementation of their best practices workflows and written them in the WDL script. So I know you've heard that term a few times now, which is WDL, which is the workflow description language. And it inside of the WDL, you can actually see the command line being used in the invocation of each GATK tool, so you can see all the details you'd like. And the nice thing about the workflow is that it combines all the steps for you, so it's a single launch of a pipeline at a time. And if you want an even easier version of those workflows to run, then you can hop on over to FireCloud, and there you will have access to some workspaces that bundle different workflows and inputs together, and they just simply need to be cloned and launched for your use and testing.
All right, thank you, Ruchi, for this overview. So now I'd like to invite, who am I inviting for this one? I'm inviting Brad, of course, Brad Chapman, who's a senior research scientist in the bioinformatics core at Harvard Chan School of Public Health, where he develops open source community-built tools through the BC Bio project. Brian O'Connor, or is he? He's right here. Brian O'Connor is the director of the UCSC Genomics Institute's computational genomics platform, where he focuses on the development and deployment of large-scale cloud-based systems for analyzing genomics data. And I'll also invite Jose Soto, who is a software engineer in the Broad Data Sciences platform as well. Please, yes. All right. Thank you for joining us today. It's pretty exciting. Brad, you play a very important role in the genomics community through the BC Bio project. It's a really important effort that is part of efforts like this to make it easy for researchers, right, to access and deploy standardized pipelines with a strong accent on reproducibility, portability. And also, one thing I find very interesting is that it enables running comparisons between pipelines, whether pipelines of the same tool but different versions, or pipelines that include different tools, sometimes competing toolkits, in a very principled way. And so what's your expectation for the GATK4 pipelines based on what you've seen so far?
It's going to be awesome. Yeah, it's no. I mean, we think one of the really cool things about the variant calling space, which is fun to work in, is that there's community built up around actually building validation sets and then tools to validate those, and then iterating on those to improve the validation sets. And so there's a wide variety of in addition to there's Genome in a Bottle, which has 3 different ethnically diverse samples, and there's new things like the like from Broad, the CHM, which is a completely orthogonal set, and this makes it easy to sort of build comparisons. And one thing we're doing in BC Bio is using those to try to validate GATK against other tools, and then iterate on those. And so, I mean, we have a bunch of, if you're interested, you can look at this after on GitHub, but there's a bunch of validations of GATK4, the beta's up. And it looks great compared to GATK3 and other tools as well. So, it's awesome that, like, on the day it rolls out, that we can start using it and recommend that people can use it.
Fantastic. Thank you for those kind words. I did not pay him to say that, though there will be food and drinks afterwards. I think the aspect of enabling comparisons is really important because, you see a lot, there's academic software toolkits, there's also commercial software toolkits out in the mix, and sometimes it's difficult to know when the authors of the tools publish like results saying, oh, my tool is better than anyone else's, which you always want to do that as a tool developer, but it can be hard for the user to really figure out, well, how was that comparison made, and was it an apples to apples comparison and things like that. And so that's why I think that something like that, for us, like we really like having a third party, an independent third party doing the comparisons, because it just takes kind of the any biases out of the equation.
Yeah, no cheating. I mean, I think one of the things that's awesome about that, Ruchi just talked about WDL, and I'm sure Brian will talk about CWL as well. It's actually making the pipeline's more open, and you can put them in lots of places. And I think that's really helping sort of open it up so that there's less divide between what you guys are doing and what someone else is doing as a third party. You can take what you ran and run it, and it makes it a lot easier.
Absolutely. Now, I'm going to call in Jose because he's been hard at work optimizing our production pipelines. So the whittles that we run are heavily optimized, thanks to Jose's work and Megan as well, Megan Shand. They've been optimizing our pipelines, and Danielle alluded to that earlier in her session, that we've been able to bring the cost of the pipelines down by a factor of 95% compared to where it was at when we started, which is huge, right? It's very big. The problem is these optimizations that you've worked so hard to put in make the scripts not portable across different platforms. So as long as they're running on Google and most flavors of Google, they're fine. But if you take them elsewhere, then some of the features are going to break. So I would say, like, what's your take on the feasibility of maintaining different versions of pipelines, and how much work has to go into testing that they're still functionally equivalent, they're still giving the same results?
Well, I think for sure, no matter any optimization you want to do, you want to make sure the science is the same. So like the most effort should be done in testing if you're going to provide optimizations, because you don't want to change the science, right? If you're messing up the tool by optimizing it, you're not optimizing, you're just making it worse, right? Like that's the bottom line. Like that's rule number one. You don't break the science when you're optimizing. But as far as optimizations go, it would be really awesome if all optimizations didn't change reproducibility or the ability to move from platform to platform. But the matter of fact is, if you want it to be as cheap as possible, you do have to take advantage of the platform you're in. Now then it becomes like a balancing act of which optimizations are worth, not being able to run on AWS, or not being able to run on Google, or not being able to run on local. And so it's a matter of like, oh, this made it 10% cheaper, 10% faster, but now you can't run on this huge platform. Like, is it really worth it? And if it is worth it, maybe so what you want to do is you want to go to the different platforms and maybe figure out like where on their platform can you optimize where you did, like in Google. Let's say we use this special option that they have to make it so much cheaper. What does Amazon have that is equivalent? That maybe not the same, but can give us the same kind of optimization that we're looking for. It's similar like the Intel optimizations that they do for GATK4 itself. It's Intel specific, right? But it's there because you need to leverage the platform you're on to make it as optimized as you want it to. Yeah. But it is a tug of war between how much better do you want it and how portable do you want it, for sure. And if we can make it all portable and all optimized, it'd be awesome. But unfortunately, that's very hard to do if you want to make it very optimized.
Yeah, and so for now, what we're going with is a compromise where we have like our production pipeline and it's the most tested. Well, that's the one that's optimized. But we do make available a generic version that we expect to be able to run everywhere. We just know that one will be. Won't have some of those gains, of course. We're willing to put some level of effort, some substantial level of effort into making those like universal versions available, because we do believe in this question of portability.
I can add one thing. I just want to add, I think everybody here, anyone who's ever had to deal with parallel versions of the same thing knows it's awful and horrible. So we really don't want to do that, but sometimes you kind of just have to, and you pay the price.
Can I ask a quick question? Do those turn on and off magically like the HMM ones do, or?
Not magically. There's some magic involved around them, but if you want, we can talk.
It'd be interesting to see if we can use the modularity. And Whittle now supports sub-workflows, and you could have like a master workflow, and then you could have a branch. If I'm running on Google, I'll run this version of this particular task, or how some things are engineered. And I think
it's very important, and it's pretty new, and not many people are using it. But I think it's a great step forward to have this portability of like 95% of your workflow, and maybe this one or two tasks are optimized for different platforms. And that'd be much lessen the burden on people.
Yeah, and if we can plug them into the workflows, I think that'll help a lot. Brian, you're involved in a lot of things. One of the many things you're involved in is GATK4 initiatives, right, to promote portability and interoperability. What do you think we can do in practice to do a better job of plugging the software and the assets we produce into the global ecosystem of tools?
Well, it was definitely interesting to hear about, the sort of like tug of war between optimization and portability, and I think there needs to be a place for both and a sort of mechanism for dealing with both of those. The cool thing is right now, like you were saying, there maybe 95% of the pipeline can be extremely portable. We have extremely good tools nowadays, right? We have things like Docker, we have CWL and Whittle. This has made our lives so much easier than 5 years ago. In the GA4GH Cloud workstream, what we're trying to do is we're trying to establish a standardized API, the tool registry service, so that you can come to me and I can say, here's my workflow. And here's some test data that goes with it. So if you're going to run it in a different platform, you can try this test data. And even better yet, here is a checker tool that will tell me whether or not that test data ran and produced a concordant result. So I think that sort of thing, identifying a standard that we can have as a community, so we can have many different repositories around the world that advertise these workflows and advertise the test data and the concordance tools. I think that's going to really go a long way to making it easier to hand tools around, hand workflows around to different people. So we're implementing this in DocStore. The flip side, I think, on this is not only to establish those standards and have places like DocStore, actually use those standards or Agora or other repositories out there, but to also actively test. And this is kind of, very in parallel with what you were talking about in terms of working on optimization and lowering cost, but on the flip side, testing portability. So one of the things GA4GH has been working on is working with the Dream Challenge folks, which has been a really nice collaboration on a series of workflow interoperability challenges. And what we're doing is the most recent challenge, which wrapped up literally like 2 days ago, we had 11 high-quality workflows, including some GATK workflows and others from GDC and ENCODE and other projects. And we paired them with test data, we paired them with a checker tool, and we handed them out to the community. And I think the last tally was 290 submissions of people running it in 17 different platforms, running these workflows in different combinations, and reporting back their successes, their failures. And this actually, I think, produced a lot of useful feedback to both the workflow engine authors and the workflow authors themselves about how do we really, as a community, make portable workflows. So I think seeing that kind of evolve in the future, seeing the repositories evolve, these challenges for showing portability evolve, and hopefully in the future we have things like, Travis CI equivalents for workflows. So as I develop my workflows, I can see, oh great, it's running in DNA Stack, 7 Bridges, DNA Nexus. I'm getting green check marks across the board. So I'm very excited by that and the evolution of what we're doing in GA4GH Dream and the GA4GH workstream.
Cool. Yeah, absolutely. I think it's a great vision for the future. Looking forward to it. Do we have questions? Do we have time for questions from our online audience?
We do not have any questions for this panel, so great job describing yourselves.
All right. Yeah, so clear. Excellent. Well, thank you so much for joining us. It's been wonderful having you here. Round of applause for our guests, please. So that's, oh, and I forgot to invite Ruchi back. You see, I shouldn't do these live things. It's so difficult. But you know what? It went quite well, I think. We had some really interesting discussions. I'm really grateful to all of the speakers and panelists who came and joined us. It's been a really wonderful afternoon talking about GATK4, knowing that it is actually out and completely finished. And oh my God, I forgot Monko. Monko, come over here. Let's do some Q&A. Wait, no, there is still one panel. I'm so looking forward to going and looking at GATK4 on the website. Please sit and join me. And my goodness, I'm never gonna do this again. I'm kidding. It's actually a lot of fun, but it's hard to keep track of. This is, going in live streaming and all of that. This is the future. I was born in 1980, so still trying to catch up. Anyway. Thank you, guys. I'm glad you were not timid and walked down before I wrapped the whole thing up. Anyway, Ruchi is still a software engineer in the Broad Data Sciences platform. And Monko used to be a Brody, but he left us. But for great new opportunities. He's now leading a research group at Yale School of Medicine, Department of Genetics, and has extensive experience in data processing in genomics for rare diseases. He recently completed his postdoc training in Daniel MacArthur's lab at Mass General, Harvard Medical School, and the Broad Institute. And so he's moving on to a new life, but hopefully still using GATK. Yeah.
Yep.
So Monko, you have quite a lot of experience with the GATK pipelines. Can you tell us your worst memory of struggling with GATK?
Yeah, so a lot of my long history of using GATKs, working on the Exome Aggregation Consortia project, which is the little brother of the Nomad project, so the project before the Nomad project. So that project went for 3 years. So when I first initially started, I had a great opportunity to work on it and work with Eric Banks, and I've seen the team change, as Eric Banks has said, how the team has evolved. This project went for 3 years, so this is my one and only opportunity to talk about the struggles over the years. So because people saw the final product, which was the release of 60,000, but they didn't see all the hard work of the GATK development team that made this all possible. So it first started of a vision of Daniel MacArthur and collaborating PIs around the world and at the Broad Institute of making a reference panel available to the world. So we started off with 13,000, then we went to 26,000, then we went to 68,000, 86,000, then the 92,000, then that was subsetted to 60,000, which was released to the world. And so I think a lot of the struggles came from being the first ever to hit each one of those milestones. And, you know, we'll find something interesting along the way, and the development team would learn new interesting things. And one of the interesting things I'd like to share with everyone is I think earlier on the days when we went for 68,000, there was assumption somewhere in the code that we wouldn't hit 50,000 anytime soon, and then we got an array out of bounds. So that was an interesting thing that got solved very quickly, but it was just an interesting point to see how quickly we were producing data both at the Broad Institute and all around the world in the other major sequencing centers. But in terms of the biggest struggle of working with a data set this large is how do you go about parallelizing this data? And none of the cloud pipelines that we've discussed today did exist at the time. So it was how do we break up those intervals such that we're sort of like partitioning the resources, the CPU resources, the memory, and things like that, so these intervals can actually run. And it was a challenge because some would complete very quickly and some would grind for 3 days. So the struggle was, one, how do you actually break that up? And the other struggle is how do you recover when some of them fail or some of them have hung? Like, how do you kill it, recover, and then proceed from then? So they were one of the biggest challenges, technical challenges we had to wrestle with the GATK pipelines back in the old days.
The old days, indeed. Ruchi, you help, as part of your field engineering job, you help researchers get started with analysis tools like GATK on FireCloud. Is there anything in what Monko was describing that kind of rings a bell and resonates in terms of the struggles that you've seen people go through?
I think even today, people do struggle with large sample set sizes and managing all that data at once and dealing with failures at different points of the pipeline and just keeping up with that. And I think, so that's hard to do, I think, on its own, and then adding on the layer of keeping track of errors that are related to the engine or any UI issues, because most of the people I work with will run pipelines through FireCloud, so their view is a little bit different. There's a few layers in between. So I think for them, the biggest struggle is trying to understand tracking errors and figuring out where the issue truly exists, be it GATK or somewhere else above it. So I think that's the hardest part as of right now.
Yeah, and it's a hard problem, and just identifying where the problem is and how to recover. Monko, you recently used FireCloud, right, to run some GATK analysis. Can you tell us how that went?
Yeah, so I've had two recent experiences over 2017, and both of them involved the Gabriella Miller Kids First project, a project that's run out of multiple sequencing centers. So this was more using the FireCloud pipeline that is very, and running a Whittle pipeline very similar to what the production pipeline looks like at the Broad, so with the help of Geraldine and the FireCloud team. So the first experience was a 900 genome data set of rare diseases with our collaborators at Boston Children's, Elizabeth Engel. And so that experience the first experience was a fantastic experience, mainly because the FireCloud team did all the heavy lifting for me, and I only had to prepare the metadata and just get the data and get it ready. So the goal of that project was to produce build 38 genomes in what we call a functional equivalence format. So a format that a lot of the major sequencing centers had agreed upon that would produce a call set very similar to each other. So where the major cost of processing data is actually the alignment. So if we can get that correct amongst the sequencing centers, that it would be a major achievement. The second one was more of an interesting learning experience for me, mainly because FireCloud team goes, well, you've seen us do it. How about you have a try this time round? So that was a 300 genome call set, again from the Gabriella Miller Kids First, a rare disease cohort. So this was more of a struggle because first of all we had to get the data from dbGaP, and that was the biggest struggle, to get the data from the dbGaP, convert the SRA to FastQ, and then it went through the FireCloud pipeline. And it was a very positive experience. I made a lot of tweaks to it, and I think that's testimony to how user-friendly the Whittle is, because the Whittle being a new technology, it's not so easy to Google. So my source of knowledge is Stack Overflow. So like if you have a Python problem or a problem in C, C++, it's normally a copy-paste problem from Stack Overflow. But because Whittle was such an easy-to-use language and easy to sort of like learn on the fly by example, it was so easy to modify an existing sort of like set of scripts to actually do exactly what I wanted to do for this time around.
I'm glad to hear that.
Yeah.
Again, I did not pay him to say that. But yeah, I think also from the support point of view, that's a big advantage I see in Whittle as well. Is I'm originally a microbiologist by training. I have no formal computational experience, and over the years, I've seen a lot of GATK users ask for help, and they have some scripts that they've put together that might be Python or Bash or something else, and it's very hard to debug when there's something wrong because sometimes it's just the level of complication of how things are implemented, whereas with Whittle we find it's a lot easier. We can make available pipelines that people can understand, like what's going on in them, and then if they make changes, it's also easier for us to follow. So that's something I'm hoping makes my job easier. Basically, it's a very selfish motivation, but I also hope that it'll help just minimize the amount of effort that people put into implementing and tweaking pipelines so they can focus on the science. Do we have questions from the audience?
We do indeed. She on Facebook asks, what is the cost of using GATK4 in FireCloud?
That's interesting in the sense of we don't charge you for using GATK or FireCloud. Those are both free to use in terms of the software that we make available. Now, FireCloud does run on Google Cloud, and Google will charge you for the compute and storage and any egress of files, so if you download data. So you still have to pay the cloud provider, but we have a free credits program that is sponsored by Google, so you can apply. It's went live today, and you can apply online on the websites and get, I think it's $250 worth of compute to get started with FireCloud and run the GATK4 pipelines.
Geraldine, I'd like to add to that, because so from my experience in using FireCloud, in terms of managing costs, my recommendation to new users out there that are using FireCloud or just a cloud platform is to actually run with some small tests. So, and instead of launching a bigger one and then getting a bill that they weren't expecting. So just run with smaller tests so you know, optimize it, and actually get it to the state that you want on a very small test case, then scale it up. Trust me, it makes conversations with your boss much easier.
That's a fair point. I should add that for the GATK4 pipelines that we're making available in FireCloud, we're also providing benchmarking numbers of both how long we expected the pipeline to run on the example, like preloaded data, as well as the estimated cost. Now, the cost can vary a little bit, but so we have an estimated ballpark that gives you, like, for this WDL script on this data set, we estimated costs about $5, $10, something like that.
Great. Another question from Shi on Facebook asks, is there a webinar on using GATK4 on FireCloud?
We have a few demo videos that Tiffany put together. We have already 2 or 3 that explain the basic operations, and we do plan to do more video tutorials to help people get started with FireCloud. Yeah.
Great, and I'm getting the blink that we are out of time. So if you did ask a question in our chat, feel free to post it to our forum. We will answer it there.
Absolutely. We will answer anything that's ever posted in the forum, and we will try to follow up on any Facebook questions as well that weren't answered today. So thank you very much for Richie and Monko for, thank you for, sorry I forgot you. All right, now we're really done. It's been a interesting and quite wonderful experience from my point of view. I hope that you enjoyed it as well, whether you're here in the room or you're watching at home or at work on the Facebook live stream. I want to just close with a few remarks. I need to get to the podium to cue the thing, hopefully without running into the furniture too much. You can tell I'm getting a little lightheaded. The release is a really big deal, so you know it's been an emotional, momentous day. So I want to thank the GATK user community who have played a huge role in getting us here, getting us to the point where we have a GATK4 version to release. There's kind of no point in making software if nobody's using it, right? And at this point, we have 55,000 registered users on the forum, and you can see kind of where they're distributed. They're all over the place. They're worldwide. I'm sure there's some who are staying up late, hopefully, to watch us, or getting very early, or will pick up the recording in a few days. We want to thank you for supporting us, for giving us a lot of feedback, for helping us get the tools to a better place. And we hope you'll try out GATK4. It's really, we think that it's going to make a big difference. With that, I also want to thank the team. I'm going to play a short video. I want to thank the teams that built the software, both GATK and FireCloud, the GATK development team, the Workbench FireCloud development teams, who are all part of the Broad's data sciences platform. It sounds corny, but it is kind of a big family. It's a wonderful experience working with these people. I want to invite anybody who's in DSP to join me down here to get a round of applause. So please don't be shy. Come on, come on, come on. Development, and also all the speakers today, everybody who spoke, anybody who traveled from out of town to come here to join us for this event. I would like you all to join me at the front. Basically, I want to show how all of this is the work of a huge amount of people who put their blood, sweat, and tears into this project. And so, wow, it's a lot of people. So yeah, please all join us. And thank you again to all our partners who came, all our special guests who joined us today. See, it's an impressive amount of people, and it's just the tip of the iceberg too. There's loads more that aren't even in the room. So to all of you guys, it's a little silly because now all the people who are applauding are at the front of the room. So let's give ourselves all a round of applause. A very successful project. Thank you. Thank you. So this concludes our GATK4 launch event. I'm going to play the video that celebrates how awesome it is to work in the data sciences platform. And you guys can all join us right after for drinks and food in the lobby. Thank you.
I started off making video games once upon a time. Then I had a ginormous quarter-life crisis and went, oh no, where's the meaning in my work?
I've been in software for a little while. I started in QA and then I was in management and team building roles. I remember my first lecture, I just felt like, wow, this would be the coolest place to work.
If you're excited to sort of work hard and learn new things, if you're excited to just be a big part of science, I feel like this is the right place to be.
Everyone I talked to said, if you're in Boston, the only place you should work is the Broad.
If you're interested in genomic discovery, the Broad really is the only place to be.
It feels better to be working on something that feels meaningful.
I get to meet new people and then get to casually say I help cure cancer for work, which is just like this mic drop in any party and then no one speaks to me again.
It's a nonprofit trying to solve the world's diseases. If that doesn't sell you on it as a starting point, I don't know, there's something wrong.
There aren't a lot of frontiers left, and this is one of them that is vast and really, really important.
It is kind of intimidating where you go, oh wow, these are people who do big, big things. And then you go, they're working with the software that I write. I better make that good.
We just called 20,000 genomes, which is pretty cool. And we're gonna do 65,000 genomes. And you can't go and look and say, well, what did this other company do? Because no one else has done it.
So all of that enormous torrent of data now has to be computed somewhere. And so we're trying to make that easier.
We take software review really seriously in our group. Everything you write is reviewed by at least one other person. And it's a great way to get much better at writing code.
There are people who've been here for years and brand new people, and they seem to really value growth in a lot of different ways.
I started off doing bench research at the Broad in one of our clinical labs. I took some coding classes and kind of started moving towards that direction. And then before I knew it, I feel like I was swallowed up by the engineering team.
I've worked in environments that are just work hard, do late nights and pull weekends. Here, despite the fact that our mission is way more important, it's so much more casual and everybody is much more kind of grounded and down to earth about things.
It's just really encouraging when your boss comes to say, how are you doing? Do you like the projects you're working on? How are things at home? And you're like, awesome, thank you for asking.
I think everyone's aware of how important all of this is, but there's also a nice balance with the rest of life. Having a drop-in meditation class and yoga and boot camp, all these other things just happening, and it's just nice to be able to have so many different kinds of resources.
The work is really interesting. The people are really great. Everyone is friendly, everyone is really smart, everyone is really excited about what they're doing.
This is a place to go and do work that is meaningful, and you are actually changing the world by enabling science to happen. And the fact that I can do that with the skills that I have without a lab coat or a PhD is amazing, and I love it.